Now we discuss about the pharmacodynamics part number 6, in which we discuss basically about the receptors, pharmacodynamics of the receptors of there are basically 4 types of receptor we discuss here. What are the main types of receptor as we can see on the screen, pharmacodynamics. The first we discuss about the ligand gated ion channel receptor. What is it? We discuss later in detail. Enzyme linked receptors, nuclear receptors, and the G protein coupled receptors. So basically these are the 4 types of receptors we discuss mainly in the pharmacology, and they are the ligand gated ion channel. It means what? Suppose that this is the cell here. Inside the cells there is here the channel is present. Suppose that this is the channel. Something is bind to this receptor. Suppose that this is the receptor and this is the ligand. When the ligand bind to the receptor, the channels will be open and there is something. Suppose that the sodium channels, the sodium is entered inside it. The sodium when enters after the ligand is binding to the receptor, these are opening of the channel and then entering of the sodium like this. That something like this is happen inside the channels. Suppose that the ligand gated ion channels. Next one is enzyme linked receptors. What happen inside this? Suppose that this is the cell. Inside the cell, some enzymes are present. Suppose that this is the enzyme, and on the surface there is a receptors are present, and these are linked with enzyme. These receptors are linked with enzymes. After that, what happened? Something is binding to it. It means the receptor through which the molecules, the neurotransmitters are binding to it, and they activating to the receptors. These receptors are linked with enzymes, and these enzymes are gets activated or stimulated, and they showing their effect in the second one, enzyme linked receptors. The enzymes are basically the linked with receptors. This is the enzyme linked with receptor. Second one. What is the third? The third is the nuclear receptor. Something happen like this. Suppose that this is the our cell inside the nucleus of the cell, nucleus, and this is the cell. Inside the nucleus there are several different types of receptors are present. Suppose that in cytoplasm also several types of receptors are present. They are binding to the some neurotransmitter and they moving inside it. They are also called as nuclear receptors, and inside the nucleus there are some different types of receptors are present. They are called as nuclear receptors. That's all. Once again, the another one is G protein coupled receptor. It is like this much. Suppose that this is the cell. On the cell surface there is this type of receptors are present. They are passing the membrane at the 7 parts. 1, 2, 3, 4, 5, 6, 7. The 7 part they are passing to the cell. Suppose that this is the 1, 2, 3, 4, 5. The 7 pass receptor. They are also called as 7 pass receptor. Which one? G protein coupled receptor, that is GPCR. And their activity are induced by the alpha, beta, and gamma units. Gamma units are present inside the cell. These are acting like this. There are the 3 types, basically 4 types of main receptors we are going to discuss, and all the other types of receptors we are going in the topics way. How the topic is induces in the central nervous system. We read about the dopamine receptors, serotonin, glutamic aspartate. These all receptors are discussing there. But now these are the most important receptors we are now seeing here for further study in pharmacology. And the next topic we start when the autonomic nervous system is the main receptor is basically here is the muscarinic and nicotinic receptors. The muscarinic receptors we studied in detail in the autonomic nervous system, and nicotinic also we studied in detail. But here we can see overview all of the receptors here. The first one is pharmacodynamics of the receptor, in which we first are going to study here. That is the ligand gated ion channel. As I told you, they are the fastest acting receptor because the ligands are binding to the receptor. Receptors are opening the channel and then molecules are going inside. The ions are moving inside. That like them. They are the first one, second one, third one, and fourth. Basically we study here 4 different types of ligand gated ion channels, in which the first we can see GABA A. GABA is having basically 3 types. GABA A, B, C, A, B, and C. So B1 is the GPCR, means G protein coupled receptor, type 4. The type B of the GABA is GPCR, and A and C are the ligand gated ion channels. Now you can tell me which one GABA is ligand gated ion channel, GABA A or GABA B? GABA A. GABA B is G protein coupled receptor. They are acting. There is a difference in action. How they acting? In the GABA A, there is ions are going inside the cell. But in GPCR, there is a GABA B receptors, the gamma aminobutyric acid, that is the GABA B, inside which there is a GPCR acting. And the GPCR how acting? The GPCR acting with the molecules are binding to it. When the acetylcholine are binding to the GPCR receptor, they are activating the alpha, gamma and beta subunits. Basically the alpha subunits are broken down and they are activating them. So that's why we can see here, now we are studying only what? Ligand gated ion channel. That's why we discussed the first one is GABA A. GABA A is only ligand. And glutamate receptors. Glutamate is having three types of receptor: NMDA receptor, AMPA receptor and kinetic acid or kinase receptors. The three types of receptors of which NMDA receptor, AMPA receptor and kinetic acid or kinase acid or the kinase receptors. There are three which type of receptor? Glutamate receptor. Next one is serotonin receptor, 5-HT3 serotonin receptor, that is also called hydroxytryptamine. They are the 7 types. They are basically 7 types, 1 to 7. They are 1 to 7, in which 5-HT3 from 5-HT1, 5-HT2, like this, 5-HT7, 5-HT3 one is ion channel. In 5-HT3 there is ions are moving inside and outside. That's why they are the ligand gated channel. All others are GPCR. Means 5-HT1, 5-HT2, 5-HT4, 5-HT5, 5-HT6, 5-HT7. These all are GPCR. There is a G protein coupled receptor are acting. Like this. We have to memorize. Next one is nicotinic receptor. Nicotinic we basically don't know now, but I am telling you. Nicotinic receptor is 2 types, NN and NM. What does it mean? NM is nicotinic neuronal receptor. They are basically present in all the neurons, that is autonomic nervous system. Nicotinic neuronal. And this one is neuromuscular. N stands for the nicotinic neuromuscular junction. They are present in the neuromuscular junction. What is neuromuscular? Hope you know. Neuromuscular junction is basically, suppose that this is the muscle and this is the neuron. And the neuron, their parts will be connected with them, that is neuron and muscles. Muscle and neurons are combined together at that region of the neurons, that is called as neuromuscular junction in simple way. What are the nicotinic receptor 2 types? NM and NN. Here nicotinic in the 2 types of acetylcholine receptor. Acetylcholine is having 2 types of receptor, nicotinic and muscarinic. Muscarinic is basically GPCR. Means they are acting through the G protein coupled receptor, but nicotinic one is only ion channel receptor. They are ligand gated ion channel action. So here we can see the nicotinic. Nicotinic one is what type of receptor? Ion channel. They conduct sodium ion. We can see here. And what is the structure of this? We can see here, I am drawing here some structure. Suppose that this is the lumen through which the ion are moving outside and inside. And aside the lumen there is a 5 flower like structures. The petals like structures will be present like this. And this is the lumen. Through this lumen, suppose that this is the cell and this lumen is located here, and there is a 5 different types of flower petals are formed inside it, and through this moving ion sodium is inside it. It means what? Moving inside or outside. But we can see here basically what happened to these cells. These are having pentameric structure. Pentameric, so which type of receptor? Nicotinic receptor is having pentameric. This is the lumen through which the sodium is inside and outside. But here we can see one more word. They have out of 5 these structures, petals like structure, these are called as pentameric structure. The pentameric structure of nicotinic receptor. What happened to this? There is a alpha unit. There is a 2 alpha unit. It means one is this one and one is this one. There are 2 alpha units, and from this alpha unit 2 finger like projections are coming towards the lumen. There is a finger-like projections are coming towards the lumen like this, and they closing this lumen. The lumen is closed by the finger-like projection of the what? Alpha subunit of the pentameric structure of the nicotinic receptor. So these are the finger-like projection. They are coming from where? Alpha subunit of the pentameric structure, and they closing the lumen. When the acetylcholine is binding, to this alpha subunit of the what? Acetylcholine is binding to the alpha subunit of the pentameric structure of nicotinic receptor. Acetylcholine is binding. These finger-like projections are moving away from the lumen. They are moving away, and they open the these lumens are open now, and through which the sodium are moving out and in. The opening and closing is regulated by the alpha subunit. Alpha subunit is responsible for the opening and closing of the lumen, through which the sodium ion inside and outside movement is regulated. It means the alpha subunit is most important here. We can see other three parts. There is this one, and this one. This one is the beta subunit. This one is the delta or eta epsilon, and this one is the gamma subunit. We can see in this picture. This is the beta subunit. This is the alpha subunit. Acetylcholine is binding to these receptors. Acetylcholine is binding to this receptor, and this finger-like projection is closing now. When the acetylcholine is binding, they are moving away. And the subunits are constant, through which the sodium are moving outside and inside. There is a beta subunit, gamma, delta, epsilon. They are not working. There is a alpha one is most important. They are closing and opening of the lumen. This is clear. Hope so this will be clear. Next point we discuss here is the pentameric structure we discussed. There is a two constant subunit, that is two alpha subunits, and remaining subunits will be the beta, gamma and delta. Alpha subunits have projection, they closes the lumen. Acetylcholine is binding to it, present in the alpha subunits. As we discussed, when the acetylcholine binds the alpha subunits, the projection moves towards the alpha subunits. When the finger-like projection moving towards the alpha subunit, and they opens the what? Lumen. The opening of the lumen, and what happened after the opening of lumen? The exchange of ions will be occur, and what is the sodium ion moves through the lumen to the different types of organ where these receptors are present. And the nicotinic receptors are what type of what type of receptor? They are the acetylcholine. Acetylcholine is binding to them, and they are gated. There is a gate is present. They are opening and closing. There the sodium ion is moving inside, and there is a channel, tunnel like a structure is present. That is the channel, and the receptors is present. Nicotinic. There is a acetylcholine gated sodium ion channel receptors are located. Next one, second part we can see here. We are going to see second part, enzyme linked receptors. They are linked with enzymes. We can see here enzyme linked. What happened inside the cell? As we discussed previously, this is the cell. Inside the cell, enzyme is present, and this is linked with the receptors. When the something is binding, suppose that this structure is binding here, and after that these are activating what? These enzymes. The inside the cell, enzymes are linked inside the cell. They are linked with the receptors outside the cell. Whenever the receptor is stimulated, the ligand, it stimulates the enzyme inside the cell. Simple. When the receptors are located outside the cells, they are linked with inside the cell with enzyme. And when the something is bind, it means the ligand is binding to these receptors. They activating what? In the early. They activating what? They activating the enzymes. And that is called as the enzyme linked receptor. This is our second discussion. Now the third one we can see here. Third one is nuclear receptor. In nuclear receptors we basically discuss they are located inside the nucleus. First we discuss enzyme linked receptors for several examples. There is a tyrosine kinase receptor is an enzyme linked receptor. Tyrosine kinase is an enzyme. Janus kinase receptor is also a type of enzyme linked receptor, and the guanylyl cyclase receptor is also a type of enzyme linked receptor, and the serine or threonine kinase receptor, kinase and cyclase, these are the all enzymes, and these enzymes through which the receptors are acting. And what different types of receptors do you know in these parts? Enzyme linked receptors. We have to memorize all these names. In the enzyme linked receptors, as we can see here, in this enzyme linked receptors, several examples you have to learn. There is a first tyrosine kinase, second Janus kinase, third guanylate cyclase, and the fourth one is serine or threonine cyclase. Now we are going to discuss first one, serine. Serine we can see here. There is a transforming growth factor. There is only one receptor, that is called as TGFR. Transforming growth factor receptor. Transforming growth factor receptor. When the anywhere written TGFR, that means transforming growth factor receptor. It is located where? It is the enzyme linked or nuclear receptor. It is the enzyme linked receptor, nuclear receptor, GPCR. What type of receptor is it? It is the enzyme linked receptor. We have to memorize it because there is MCQ questions are coming from there. What type of receptor is it? TGFR is what type of receptor? Nuclear receptor, or what type of receptor is it? So we have to memorize these are the enzyme linked receptor. Simple. Next one, guanylyl cyclase. There is a ANPR and a BNPR. We have to memorize atrial natriuretic peptide and the next one, brain natriuretic peptide. A stands for atrial, B stands for brain, N stands for natriuretic peptides, and P stands for peptide. There is a atrial natriuretic peptide, that is ANPR, and the BNPR is what? Brain natriuretic peptides. These are acting on the blood vessels mainly. These are acting on the blood vessels. They are causing, their effect will be the vasodilation. The blood, suppose that this is the blood vessels hole, their vessels will be vasodilation. Their whole diameter will be increases. It means the blood vessels gets dilated. They are dilated. It means dilation is done by the which type of enzyme linked receptor? That is the ANPR in the and the atria of the heart, and the BNPR in the brain. That is the increases through which type of coenzymes or the secondary messenger. The secondary messenger acting here is the cyclic GMP. Cyclic GMP activity will be increased. How they increases we discuss later. Now we have to memorize that they are acting as a vasodilation. They are acting mainly in the located in the blood vessels of the brain and blood vessels of the heart. We can see atrial natriuretic peptides receptor and the brain natriuretic peptides receptor. Next one is Janus kinase receptor. JAK-R. JAK-R means Janus. A stands for the Janus kinase. Kinase is an enzyme. They are the basically types of tyrosine kinase receptor. They are the subtype of tyrosine kinase receptor, and before it we have to study tyrosine kinase receptor. We can see here. They are the first one is EGFR, that is epithelial growth factor receptor, and they are also called as HER1, and their second one is HER2 receptor. The HER1 is epithelial growth factor 1 receptor, that is HER1, and second one is the HER2. Next one is VEGFR, vascular endothelial growth factor receptor. Vascular endothelial growth factor receptor. They are also type of tyrosine kinase receptor. Next one is insulin receptor. Most important. They are also the hormonal receptors, or the enzyme linked receptors. They are also under the tyrosine kinase receptor. Their action. And the IGF-1, that is the insulin-like growth factor 1 receptor. These all are coming in pathology or the different topics in the physiology. So we have to memorize it. What different types of receptor are linked with the enzymes. The enzyme linked receptors are the tyrosine kinase, Janus kinase receptor, guanylyl cyclase receptors, and the serine or threonine. In Janus kinase receptors, we have to memorize basically growth hormone receptors are the enzyme linked receptor. Cytokine receptor, most important is prolactin receptor, is also is a type of enzyme linked receptor. So what we know now, till now we study about the enzyme linked receptors. What are the four main types of enzyme linked receptors? Tyrosine kinase, Janus kinase, guanylyl cyclase kinase, and the last one is serine and threonine. To memorize all the names and their actions is also memorized. Next one we are going to study about nuclear receptor. Nuclear receptor is the slowest acting receptor because they have to any neurotransmitter have to cross the first inside the cell. After that, they inside the cell reaching into the cytoplasm, they are entering inside the nucleus of the cell, and after that they are acting. That's why they are the slowest one, and they are the slowest acting. The location of this is basically both in the cytoplasm or the nucleus. Some of the receptors are located basically inside the cytoplasm. After they are binding to the ligand or the different types of neurotransmitter, they are reaching to the nucleus. That's why there are the several Some of them are present inside the cytoplasm also, but after they binding to it, they're reaching into the nucleus. That's why they are also called as nuclear receptors. And some of them are basically present inside the nucleus. They are not present inside the cytoplasm. They are present directly inside the nucleus. They are the basically two locations, nucleus. Some of them are present inside the nucleus, some are present inside the cytoplasm. Basically, these all are actions inside the nucleus, but some of them are present inside the cytoplasm, as we discuss now. In the thyroid hormone receptor, thyroid hormone basically T3, T4 receptor. Next one is vitamin A receptor. Vitamin A is RAR and RXR. Two types of receptor of vitamin A, RAR and RXR. RAR is the retinoic acid receptor and the RXR is the retinoic X receptor. The RAR is retinoic acid receptor and RXR is the retinoic X receptor. So these are the two vitamin A receptor. These are also present on the nucleus of the cell, and the T3, T4 thyroid receptors are present inside the nucleus. Next one we can see PPAR, that is paroxysmal proliferative activated receptor, paroxysmal proliferator activated receptors. Next one is estrogen receptor, and the last one is progesterone receptor. So these are the 5 receptors which are located inside the nucleus. You have to memorize all these names. These are important. The thyroid hormones receptors are located where? Nucleus. Estrogen, nucleus. Progesterone, nucleus. PPAR, that is nucleus. RAR, RXR, nucleus. Vitamin A receptors are located nucleus. Next one is the present in cytoplasm. What are the receptors are located inside the cytoplasm? In the cytoplasm, basically they are located is the androgen receptor, mineralocorticoid receptor, glucocorticoid receptor, and vitamin D. This is important. Vitamin D receptors are located where? They are also located inside the cytoplasm. Vitamin A receptors are located directly inside the nucleus. But after the binding to the ligand or the neurotransmitter, they are enters inside the nucleus. That's why their location will be important. The examples are also important here. You have to memorize. Next point is the G protein coupled receptor. As we discussed previously, suppose that this is the cell membrane of the any cell. On this, there is a several different layers of these receptors are moving like this. As we count, like the 7 times they passes the membrane like this in the zigzag manner, and that's why they are called as 7 time receptors they passing. That's why they are called as 7 transmembrane receptor, or the 7 pass receptor. They are also called as hepta means 7, hepta helical like a structure moving, that zigzag, that is hepta helical structure. Next one is serpentine, they are moving serpentine, a snack like serpentine structure, or the serpentine receptor. Next one, metabotropic. This term is important. When the ligand get ion channels, these all are called as ionotropic channels, and when they are connected with G protein or the GPCR, they are called as the metabotropic receptors. The metabotropic receptors, these all are the 4 names. These all are the 4 names of GPCR. The GPCR is having 4 names. What are them? That is a serpentine receptor, 7 pass receptor, heptahelical receptors, or the metabotropic receptors. What happens when the drug is binding to these receptors? We can see here. First, the receptor through which the positive responses happen means drug is bind, and there is the 3 subunits, alpha, beta, and gamma. We can see in this alpha, beta, and gamma. They are the 3 subunits. These 3 subunits are in resting stage. Resting state means in the cell is in resting condition. There is no any stimulation of cell. When the any receptors through which the ligands are binding to these receptors, they activating. Suppose that the acetylcholine binding to them, and the metabotropic, basically M1, M3, M4, M5 receptors are present, muscarinic receptor, and they are binding of the acetylcholine. They are activating these serpentine receptors, and the alpha, beta, gamma subunits will be activated. And the alpha, basically alpha 1 is most active, and the alpha 1 gets dissociate from the beta gamma. Dissociate means breakdown. How they gets breakdown? There is a GDP molecule is present. We can see here GDP. Let me clear GDP. The GDP molecule is attached with the alpha subunit. When the phosphate is coming near to the serpentine receptors after the stimulation, these are more greedy for the phosphate, that is GDP with the alpha subunit, and they attach to they forming GDP is combining with 1 phosphate is from GTP. That is definitely 2 phosphate is combining with 3 phosphate, they forming GDP to GTP, guanosine triphosphate. With the alpha subunit, they are dissociated with alpha subunit, they are more greedy, and the beta and gamma subunits are alone, they are dissociated alone. It means the GTPase enzyme activity will be done here. GTPase, through that GTPase enzyme, the GTP is converted into the GTP. After that, this cycle, they are get phosphorylated. The alpha subunit will get phosphorylated, and once again they loses the energy and they converted into the phosphate and GDP. This type of recycling will be happened here in this way. And next one, we can read what we discussed above. We can read here: the receptors passes through the cell membrane 7 times. The receptors then bind to the 3 proteins alpha, beta and gamma. The alpha subunits to the GDP bind. In the presence of phosphate, the GDP is converted into GTP. Alpha subunits is separated from the beta and gamma. They moves with the GTP, and the 8 alpha has GTPase activity. The phosphate is removed by the breakdown of GTP. As we discuss everything above, which is written here. Now next point, we can see alpha subunit is most important, and alpha subunit is having 3 subunits. Alpha subunit is having 3 subtypes, that is GS, stimulatory, GQ, quiescent and G naught. They are the 3 phases of the alpha subunits is present, that is the GS, GQ and G naught. How the GQ and GS means? The GS means G stimulatory. They are acting through the enzyme, that is adenylyl cyclase, that is alpha GS is the stimulatory. They are acting with alpha stimulatory with the GTP, and their enzyme is adenylyl cyclase, same as GQ. GQ is acting with GTP. The enzyme is phospholipase C, and same as we can see third one we study first to this. There is a subtypes of subunits, that is GS and GQ. We can see here examples: adenylyl cyclase and phospholipase C. The adenylyl cyclase is activating inside the cell. Suppose that cell is like this: serpentine receptors, alpha subunit. Alpha subunit is activated cyclic AMP. The cyclic AMP concentration will be increases, and they activating a protein kinase A. Protein kinase A is activated, and they phosphorylate the muscles. They phosphorylate the proteins, and after that the action will be, we can see that these things are happened. We can see here what happened after that. The alpha subunit is activating the enzyme adenylyl cyclase. The adenylyl cyclase enzyme is broken down, and they producing the cyclic AMP. And after that they stimulate the cyclic AMP is stimulating the protein kinase A, and where the protein kinase A is basically stimulated, we have to know where the protein kinase A is stimulated. So these are stimulated basically inside the smooth muscle and in cardiac muscle and skeletal muscles. So there are 3 places where they are stimulated. The first one is cardiac muscle, a smooth muscle and a skeletal muscle. In smooth muscles, what are the enzymes they stimulated? They are the MLCP and MLCK. There is myosin light chain phosphatase enzyme gets phosphorylated. Phosphorylated means something gets converted into new formation. After that they are the raw material for them. They are converting to them. And next one is MLCK, myosin light chain kinase. One is kinase and another one is phosphatase. These both enzymes are phosphorylated by the protein kinase A in the smooth muscles. What happened in the cardiac muscles? In the cardiac muscles they activated the calcium channels, and they increase the calcium channels is phosphorylated. It means the calcium channels are firstly inactivated. They are gets activated, and they opening the calcium channels. The calcium channels are more excited and through in the muscles, and they leads to contraction. The calcium channel is responsible for the contraction in the muscle. For example, beta 2 receptors in the cell. Beta 2 receptors, where they are located in the smooth muscles, cardiac and the skeletal muscles. We discuss later where they are located and what are the locations. Till now we have to memorize GS means alpha S. Stimulatory activity is done where? In the smooth muscle, skeletal muscle and cardiac muscle. Where the all done in the smooth muscle basically is done by the smooth muscle is done by the MLCP and MLCK. Myosin light chain phosphatase and kinase. In cardiac muscle, calcium channel is activated. Next one we can see here GQ, that is alpha quiescent stage is activated, and there is a phospholipase C enzyme is activated inside it. This phospholipase C enzyme is forming the PIP2, phosphoinositol bisphosphonate or the phosphate, and these are breaking into 2 forms, IP3 and DAG, diacylglycerol and inositol triphosphate. They are broken into two parts, inositol triphosphate and diacylglycerol. The diacylglycerol is basically present on the membrane of the any protein kinase, and this one is activating, the inositol triphosphate is activating the calcium channel and the alpha 1 receptors. They are basically activating the alpha 1 receptors and calcium channels, and DAG activating the membrane protein kinase. What does it mean? What we are discussing here, suppose that this one is cell. In this cell the GQ stage is activating. It means there is a enzyme phospholipase C, they acting on the cell membrane and breaking down this cell membrane. Lipase means cutting down and the breaking down, and they forming the PIP2 phosphoinositol phosphonate to bisphosphonate or the biphosphonate. They are converting into it, and after that they break down into the DAG and IP3, inositol triphosphate and diacylglycerol. This inositol and triphosphate is acting. We can see here the inositol triphosphate is acting on the calcium channel of the cell, and also on the alpha 1 receptor. Well, the alpha 1 receptors are where it is located, we discuss later, but we have to memorize the alpha 1 receptor action is done by the GQ IP3, which part? Calcium channel activation, and that leads to contraction. Another one, DAG, is activating the membrane phosphorylation, membrane protein kinase. We have to action like this. So this is all about the GQ and GS. Now the next one type is we can see here one drug, salbutamol. Salbutamol is acting on the beta 2 agonist drug. They are acting, and there is a smooth muscles contraction or the bronchodilation in the cardiac muscle or the skeletal muscle. They cause palpitation, tremors. We discuss later it in the sympathetic nervous system. Leave it for now. In subtypes of alpha subunits, there are basically three types of subtypes: GI, G0, and G12 and 13, 1 and 2. The GI is inhibitor and GO is other different types, and G12 and G13 we can see. In the GI or inhibitor, alpha segment will be inhibited, and they increasing the GTP. As we can see, inhibitor means they're decreasing the cyclic AMP. What they are activating? They stimulating the cyclic AMP basically in the stimulatory phase. It means inhibitory phase they're decreasing the cyclic AMP, and this alpha inhibitory is leads to phosphorylation of potassium channels. Potassium channel phosphorylation means opening. Phosphorylation means basically there gets activated. So they opening the potassium channels and cell gets relaxed. Why? Inside the cells positive charges are present. When the positive charges are loses, the cells are gets in inhibited state and they gets relaxed. Suppose that G other type means beta and gamma subunits are gets activated. Beta and gamma subunits are gets stimulated after the breakdown of alpha and beta gamma segments. The beta and gamma segments are stimulated. When they gets stimulated, they are decreasing the calcium channels. Calcium amount will be decreases because they opening the potassium channel. They also open the potassium channel, and that leads to relaxation. It is okay. Where they located these type of channels? In M2 receptors. M2 receptors, where it is located we discuss later. In parasympathetic nervous system. M2, it means muscarinic type 2 receptor. They are located whole throughout the body, and inhibitory action on the heart. The inhibitory action on heart and stimulatory action on throughout the body. We discuss whole these activity detail in the sympathetic and parasympathetic nervous system. Now the G12 and G13, they are basically activating the Rho factor. The Rho factor is different. They are activating the Rho is two type of action, GTP with and kinase with. The Rho at GTP and Rho kinase, they leads to the vasoconstriction in the smooth muscle contraction. Basically in the smooth muscle, the contraction is done by the two types of activity, that is the G12 and G13, and they are activated by the GTP and G kinase. There is a Rho kinase activity drugs will be done, that is Fisardil and Natrasodil. We discuss later in the central nervous system drug and the eye drug later, not now. For now, that's all. I think it will helpful for you, all these receptors. We have to memorize first the name of receptors. Later on we have to memorize the action, how they acting on the receptors. These all we discuss later in different parts of the segment of our pharmacology. So don't worry. And